Okay, so we have talked fairly theoretical about tree construction or use some minor examples. The reason the question then is what happens when you actually create these kind of trees with all kinds of living things? What's the result in reality of this tree building process? And that result is shown right here. And you've seen this before. This is the three-domain tree. And so when you do this, you find that organisms fall into three major branches, the ones we now call the bacteria, the archaea, and the eukarya, where these are the nuclear lineages of the eukaryotes. There's some nomenclature issues with this. This is sometimes called the three-kingdom tree, but historically, the kingdoms included, plants as one kingdom, fungi as another kingdom, animals as a kingdom. And so rather than try to change that notion of what a kingdom is, a higher level of taxonomy was invented called the domain. And so each of these is a domain comprised of independent kingdoms. Now, confusingly, it's very common for these different branches of bacteria to be called phyla, not kingdoms. And so there's some, if you look, for example, on the NCBI website for taxonomy, you'll notice that a lot of organisms don't include all levels of the standard Linnaean taxonomy. That's just the way it is. It would be nice if it was better organized than that, but it's not. Yeah?
So a while ago, the two branches of the line were distinguished by Eubacteria and Archaeobacteria. Why did that change?
Yeah, so when this was originally discovered, the proposal was to call these eubacteria, or true bacteria, and these archaebacteria, that is, ancient bacteria. And for a while, those names were used. They're discouraged now for the kind of obvious reason is that what's the difference between bacteria and eubacteria, right? If there's true bacteria, where are the false bacteria? Well, there aren't any, because they're not bacteria. And these is ancient bacteria, archaebacteria, ancient bacteria. But first of all, they're not bacteria. And secondly, they aren't ancient either. Every sequence that was analyzed to create this part of the tree is a modern sequence from an organism that was alive the day before the DNA was extracted from it. So they're not ancient bacteria. They're primitive, but they're not ancient. And so those names kind of reinforce the old ladder of life notion about how these organisms were related, and so they were dispensed with. Does that answer your question? Good. And the word eukarya, or true nuclei, was retained more or less for historical reasons. So these organisms down here were known well before this organization was discovered, and were scattered amongst the bacteria that they weren't related to. There were clues that they were different. There were some real weirdnesses, particularly on membrane lipids, but this was before the time when a lot of genetic analysis was done, before a lot was known about transcription and translation and so forth in these organisms. So they hadn't really been studied that well prior to this, and their differences weren't clear. Now they're quite clear, and we'll talk about them later. You've probably talked about them already as well. So there are three kinds of living things on Earth. But you'll notice that they are all connected. There's every reason to believe that all known living things on Earth have a common ancestry. You can imagine places or scenarios where this wasn't the case. But all of these things, I mean, the fact that you're doing ribosomal analysis to get this tree tells you they all have ribosomes, right? And that really by itself is enough to tell you they have a common ancestry. Another big outcome of this is that big eukaryotes are really a tiny part of biological diversity. So here, looking back at this tree, here's Homo representing animals, Saccharomyces representing fungi, and Arabidopsis representing plants. These the tips of these three branches are all of the macroscopic world that we normally think about. These are three of the 5 kingdoms in the old 5-kingdom domain system, the 5-kingdom tree. They represent a tiny fraction of the real biological diversity. Most of biological diversity is in these other organisms. Bacteria, to a lesser extent, archaea, and things that people used to call protists or protozoans. One of the outcomes of this is, if you look at these old geologic scales of living things and of rocks and so forth, one of the things you'll often see is that eukaryotes emerge in the fossil record at about 2.2 billion years ago. That is about halfway through the history of Earth. This is clearly not the case. These organisms, the root of this tree is somewhere in here. We'll talk about that later. The eukarya are essentially as ancient a group of organisms as either the bacteria or archaea. So stop thinking of eukaryotes as some modern advance on these other organisms. That's not the case. They have their own emergence at about the same time as the archaea and the eukaryotes. An interesting thing is that this long branch here connecting, the root's going to be in here. There aren't any known really deep branches in the eukaryotic tree. There's this long branch that we don't know anything about before we start to get to the diversity of organisms we do know something about. This represents a lot of evolutionary history and change before this diversification. There are a couple of reasons why this might be. One, it may just be that there's an awful lot of evolutionary history before a radiation of surviving eukaryotes. But you'll also notice, if you look at this particular, that these deeper branches are represented by parasites. Giardia, cause of Montezuma's revenge. Leishmania, which you probably have heard of. Probably the reason for that isn't that those are representative, really, that all the organisms that branch off down here are parasites. It turns out that there are any number of protists swimming around, particularly in anaerobic environments, that you can see and characterize, but that hasn't been done. Where they haven't been named, they haven't been studied, nobody knows anything about them. And the reason is because we don't have a lot of reason to study them. Whereas parasites, we want to know everything about. And so we have this strong incentive to study parasitic unicellular eukaryotes. And we've done that, and the result is we know more about the diversity of these organisms from the parasites than we do from the free living organisms. I think that'd be very interesting to see what kind of creatures exist down in this part of the tree. The reality is that probably the eukaryotes, as a large group, are less well understood than either the bacteria or even the archaea. There's a lot of diversity in here that we don't know very much about. The bacteria are not divided into two groups, gram-negative and gram-positive. And so this is a tree of bacteria, and this isn't quite correct. This part of the tree shouldn't be read. There are two main kinds of gram-positive bacteria, the firmicutes and the actinobacteria. Firmicutes are organisms like bacillus and clostridium and staph and the lactic acid bacteria. The actinobacteria are essentially the acid-fast bacteria, things like Mycobacterium, Corynebacterium, and so forth. Gram positivity had an independent origin in these two groups. There are members of both of these that branch off deeply down here that have a standard issue gram-negative cell envelope. The firmicutes lack an outer membrane completely. Most of them, the ones we're familiar with. Again, there's a bunch of them that branch off down here that have an outer membrane. But the well-known firmicutes lack an outer membrane completely. The Actinobacteria, the well-known ones again, do have an outer membrane. But that outer membrane is made of mycolic acid, not phospholipid. The commonality here is just that when you soak them in crystal violet, it sticks really well. That's it. That's all it means. These two groups, the chlamydia and planktomyces, also lack an outer membrane. But they don't stain gram-positive, and so they're not usually considered to be gram-positive in the usual sense. So there is no gram-negative, gram-positive division in the bacterial world. Now that doesn't mean that it's not a completely useful way to characterize organisms, in the same way that if you're walking through the woods and you see a purple flower, that purpleness is useful information to know about. But it doesn't have anything to do with the phylogenetic relationships of the creatures. Lastly was proof of a long-standing hypothesis called the Endosymbiotic Hypothesis. The notion that some organelles in eukaryotes are the remnants of bacterial endosymbionts. This is clearest in the case of plastids or chloroplasts, where in some organisms they look an awful lot like cyanobacteria. And the reason for that is because they are cyanobacteria. This kind of endosymbiosis is really, really common, particularly for phototropes. There are any number of unicellular eukaryotes that take up cyanobacteria and keep them inside, and they are functionally chloroplasts. But they can be released or taken in at will. They're not obligate symbionts. Even animals can do this. How many of you have heard of coral bleaching? What's happening? Those animals, the corals, take up symbionts, photosynthetic symbionts, and they function just exactly like chloroplasts in these animals. And so most of the energy that a coral reef acquires is through photosynthesis in animals. And when those symbionts are lost, the corals lose their color, most of them, and the animals can't survive. They can't obtain enough energy using the traditional methods, using heterotrophy. So that kind of thing happens all the time. Mitochondria are also the relics of bacterial endosymbionts, in this case of alpha proteobacteria. And although the details of it have been a little bit hard to sort out, the relatives of mitochondria seem to be those alpha proteobacteria that form intimate symbioses. For example, rickettsia and Bartonella and so forth. These are endosymbiotic alpha proteobacteria. And that process seems to have become permanent. Now, these organelles have gone through a huge amount of evolutionary change since then. They have incredibly high evolutionary rates. So that relationship is a little hard to sort out. How do we know this? How does this tree prove this? It proves it because these organelles have their own DNA, and those DNA include ribosomal RNAs. And so, for example, in the case of Saccharomyces, if you were to do this tree completely, actually occurs twice in the tree. Once here for the nuclear gene, and once over here for the mitochondrial gene. And an organism like Arabidopsis appears 3 times. Once for the nuclear gene, once for the mitochondrial gene, and once for the plastid gene. Did you guys ever learn anything about genetics in mitochondria? No? It's a nightmare. I mean, it really is. you realize that in mammals, there are only 2 transcripts in the mitochondrion? One goes this way, one goes that way. And all the genes in the mitochondria are processed out of those 2 mRNA, primary mRNAs. You know how that processing takes place? RNase P. I told you about that earlier. Every one of those open reading frames has a tRNA between them, and they're processed out. And the mRNAs are processed from that. What a headache. Now, this notion had been around for a long time, at least 100 years, based on observations, microscopy. It was championed by, what's her name? I can't remember her name, Carl Sagan's wife. I don't remember her name. Anyway, she championed this notion in recent times. The notion that she also had was that there might be other organelles in eukaryotes that were the remnants of bacterial symbionts, particularly the microtubule cytoskeletal structures, the basal bodies of cilia and flagella, the centrioles and spindle apparatus. These bear, particularly in some protists, pretty striking resemblance to spirochetes. But that resemblance seems to be superficial. The proteins involved aren't homologous, and those organelles don't contain their DNA, so the hypothesis can't even be tested. There's no evidence for specific spirochete genes in the nuclei of eukaryotes, except for one tRNA synthetase, which clearly is not related to that.